kind of a side project that I work on. I don't normally study salty systems. This is kind of like a side pet project that we have going on until my other samples come in next year. So why do we care about salt flats and astrobiology? Because we know there are salt flats on Mars. These are the salt flats that were studied in 2005, and they're thought to be a remnant of a large body of water that used to be in that area a long time ago. And these are the salt flats in Utah that I'm currently working on that were also where Lake Bonneville used to be. And then when Lake Bonneville evaporated, we were left with the Bonneville salt flats. And so there's a couple very key things between these two places. One is that the salt flats in Utah undergo these ephemeral wet-dry cycles. And so right now and all winter long, they were covered in a layer of water. And then as it heats up and warms up, the water evaporates off and dries out, and then you get this salt crust formation. There haven't been too many studies on the salt crust formation and what are the biophysical chemical properties that lead to it. There have been some geology studies where they've gone and drilled cores and looked at like kind of what are the mineral components on these salt flats. But the only microbiology studies that have been done to date are culture dependent. One of our collaborators has gone out and tried to grow things from them and isolated your standard Haloarchaea, some Selenobacter, things like that, that we know are in salty systems and aren't very interesting from my perspective. So when we culture things, we only get like 1% of the microbial community. So we're taking a culture independent approach and looking at what are the total anaerobic and aerobic microbial communities out there. And then I want to dig a little bit deeper and sort of ask this question: what are they doing, and what are the species that are really responsible for the primary carbon fixation in the system, and do they need hydrogen as the electron source? And so this second question sort of comes more into play when we think about the anaerobic community members that could be fixing carbon in there, and more specifically the methanogens and the acetogens that might possibly be there because they both need hydrogen as electron donor. And so the Bonneville salt flats are also an interesting space aside from other salt flats because you have a lot of human activities that go on out there. So these high-speed land races take place every year that the salt crust thickness is actually thick enough for them to occur. And so you have like the highest speeds that have occurred on land have been out here, and also the highest speeds on a bar stool have taken place there. And so we also have mining processes that are going on there. They pump out the underground water and pump it into evaporating ponds and then sell that as table salt. So you might be eating salt from Utah with your dinner tonight. Okay, so we went out and sampled some things as we do as environmental microbiologists. So we paired up with our geology collaborators, and the geology took samples all over here from every single site labeled. They actually used this really cool drill and drilled them out, and they got down there fairly deep. However, all of these samples were useless to us because they collected them in a way that was not aseptic. There was no way to drill them cleanly. So we went out and instead dug pits. So we sterilized our tools between sites as best as we could with ethanol and flame. And these are the sites that we sampled in red right here. And so this black line right here is the racetrack. And so we tried to get a sample on and off the racetrack and sort of in this transect across the entire area. And these are what our pits look like at each one of those sites. So you can see in this site right here, oh, these aren't really coming up too well. Well, I'll just tell you that there were textural layers as you got down. And so they looked different. And we split them up according to those like visual and textural layers. Some of them had only 2 layers, and some of them had 3 layers, and one of them had 4 layers. And so we got back to the lab with all of these samples. homogenized them, extracted DNA from them, sent a subset off to geology to do mineral analysis. And then we sequenced them using 16S sequencing, and we will be doing metagenomic sequencing on a select few of these pits. We haven't decided which ones to do that yet. That's kind of the next step. And I also set up these enriched carbon incubations to try to get at this question of what are the microbes doing there. So I wanted to dig a little bit deeper beyond who is there and try to answer this, what are they doing? And so I used stable isotope tracing with 13C carbon. So these are all of the media that I set up incubations with. I chose glucose to sort of get an idea of what the heterotrophic community is like. So what are the organisms that are consuming carbon produced by other species there? I chose acetate because I'm interested in the interplay between acetogens, organisms that make acetate, methanogens, organisms that make methane, and the heterotrophic community. So can the heterotrophic community consume the acetate made by the acetogens? And I set up these bicarbonate incubations also to sort of get at this play between methanogens and acetogens. They both usually compete for CO2, as well as even sometimes you have sulfate reducers that can use all of these compounds. And so sulfate reducers will generally outcompete both acetogens and methanogens for things. They're just much better at grabbing these things, and it takes them much less energy to do what they're doing than the methanogens and the acetogens. So that's why I did all of these. And then we had one with hydrogen and one without hydrogen. Again, this is because the acetogens and the methanogens specifically need the hydrogen as electron donor, as well as bicarbonate to grow. And so these are the incubations right here. They hung out on my bench for about 60 days. And then I extracted all of the headspace inside of these exotainer tubes and split that up into 2 samples for the glucose and the acetate samples, one to be sent off for 13CO2 analysis, one to be sent off for 13CH4 analysis. And so the reason we did this is because if you have something consuming glucose that is labeled with this 13C, they'll respire it out as 13CO2, and so you can catch that in the headspace. And likewise with methane, if you have active methanogens in your community that are using any of these 13C compounds, you can see that as a tracer in the headspace gases. So this kind of answers the question of what is the total microbial community doing, but doesn't get at the individuals that are doing it. And so I'm also doing these single-cell Raman sorting experiments using a microfluidic device that I'm currently in the process of designing. And I'm not going to talk too much about this because I don't actually have any data or results to show you guys, or even a device to do them with. But the idea is basically you can shine a Raman laser on a single cell, and if that single cell has incorporated 13C into its biomass, you'll see a shift in the spectra. So you can then take that single cell, sort it off to the side, sequence it, and answer the question of what cell was using these 13C carbon compounds. Okay, so I will talk to you about the headspace data because I have some of that back. So this is all of the data that I have back in a really complicated chart. We'll just kind of go through it step by step. So we'll look at the methane data first. So as I told you, methanogens need hydrogen to grow. So we would expect methanogens to be only producing methane in this control right here with hydrogen. However, we can see that there is some methane production in these other incubations without hydrogen. This kind of indicates that methanogens might be using hydrogen, but they could also be using something else for their electron donor. And so we did aerobic and anaerobic incubations. We're not really expecting any sort of methane production in these aerobic incubations because methanogens are anaerobic organisms and oxygen generally kills them if they're exposed to it for a really long time. So this is kind of what we expected to see across the board, either in one of these two However, in this one pit, 56, we did get some methane production in the aerobic incubations and not the anaerobic incubations. So this is very strange, and I don't really have too many ideas of what's going on here, especially because you can see in layer 4, you have a lot of it, and maybe they were consuming all of the oxygen that was in the tube, or maybe other microbial community members were consuming all the oxygen in the tube really quickly, and so you get to that anaerobic state fairly fast. I didn't set up anaerobic incubations for that layer because our koi chamber sort of crapped out on us that night and refused to work because it had no gas. So I wasn't able to set up ones for that layer, unfortunately. But also, I had a little bit of methane production right here in this layer 2. And as you'll note up here, we also had methane production from glucose, which is very strange because methanogens can't use glucose. They can't even transport it inside of their cell. And so because this occurred in aerobic incubations and not the anaerobic incubations, this kind of suggests that there's some sort of partnering going on with the aerobic microbial community members and the methanogens. And so we can kind of extend this into pit 12B, where I also saw the same thing. There was methane production from glucose going on under aerobic conditions, but not anaerobic conditions down here. And so this was kind of a strange result. But it also kind of makes sense when you look at the CO2 data. So in these same tubes right here, where I have methane production from glucose, we know that there's labeled CO2 in there. So I kind of have even more evidence that these aerobic community members are aerobically respiring glucose, turning it into 13CO2, and then the methanogens are able to use that 13CO2 in making methane. And I think a similar story is happening with acetate right here in pit 12B. And so some methanogens can directly use acetate to make methane. However, I don't think that's going on because if we look at the, whoops, let me go back. Okay, so if we look at the anaerobic ones down here with acetate and, yeah, so if we look at the ones down here that are anaerobic with acetate, there is no methane production. So methanogens are anaerobic. If they were able to use acetate directly, we would expect to see methane production in the anaerobic controls also, but we only see it in the aerobic ones. So this is further evidence that there is, in fact, some sort of interaction between the aerobic community members and the methanogens, especially in this layer 2 right here. Okay, so oops. Let me go on to what's going on. Oh, okay, so that was what I just mentioned, where you have so you have no methane production right here from bicarbonate also in layer 1, right here. Yeah, so in layer 1, you have no methane production in the anaerobic controls from bicarbonate, but we kind of see it as we go deeper in the pits. And so this is a result that sort of makes sense because you would expect methanogens to be deeper down in the soil where they're kind of isolated from that oxygen that's getting to them. Okay, and then lastly, we'll kind of look up here at what was going on in the aerobic bicarbonate incubations. And so you can see there's a little bit of methane production in that first layer. So for some reason, there were a little bit of methanogens that maybe were hanging out in here, but they can't really do anything because they're not doing anything right here when it's anaerobic. They can't really do anything unless they have their aerobic partners. So this pit right here, definitely there's some interesting things going on, and I don't really have too much else to say about that other than there's some evidence that it's happening, but I don't really know too much more until I dig a little bit deeper into this story. Okay, so who are the microbial community members? So this is our total 16S sequencing of all of the archaeal community members, and from sample to sample, layer to layer, it all pretty much looked like this. And so it's mainly dominated by Halobacteria ace, which isn't too surprising. They're from salt flats, so layer 1 to layer 3, you just get a bunch of Halobacteria ace. And these are the heterotrophic Haloarchaea that we just heard to talk about, except they're not cold adapted. They're kind of hanging out in normal temperatures. Okay, so I looked at the similarity between these samples. Even though they kind of looked all the same, when I just look at who's there, this is an MDS plot, and so the axes don't really matter too much. They're largely arbitrary. It's sort of like a PCA plot, if you're familiar with that. And it's really just the distance between each of these points that have any meaning on here. And so some interesting trends sort of come out when you look at the archaeal community in this way. And you can see all of these circles on here are layer 1. All of the triangles on here are layer 2, and all of the squares are layer 3. So you kind of have like this general shift in your microbial community as you go down. And just to kind of show you, I don't have these labeled. I just told you what was in there. These are all just down to the genus level, different Halobacteria ace. But it's not really shifting in who's there. It's just the abundances that are shifting as we go down in layers. Okay, so bacteria. So bacteria, mostly Selenobacter and some unclassified bacteria where the 16S sequences didn't really line up to anything in the database. But it's pretty much dominated by Selenobacter. Again, not too surprising. We would completely expect this. You can't see it too well, but we have some sulfate reducers right here. And we do actually find some acetogens, the acetotherma, right here. Oh, I forgot to point it out on the archaeal graph, but we did find a tiny amount of methanogens. So this is really interesting because, as I was hoping, we do have an environment where acetogens, methanogens, and sulfate reducers are all coexisting. And so I want to dig into a little bit deeper why they're not outcompeting each other and why they're all able to coexist in these sort of lower abundances in this community. But I don't really have an answer to that yet. That's future work. We'll be looking into that a little bit more. And so we can look at if we have this same shift that's going on in the bacterial community like we did with the archaeal community. And again, we have circles are layer 1, and the triangles are layer 2, and the squares are layer 3. And so again, we see sort of a little bit more muddled in the layer 2 and layer 3 with the bacteria sequences, but there is still like a general shift sort of away from layer 1 to as you go deeper into the more anaerobic community members. And here's another plot, and so this is genus level again, and it's just kind of showing that it doesn't really change. The same colors are between all of the sequences. It's really just the sizes of what's there. So the abundances of the bacterial community are changing, not exactly the individual sequences themselves. Okay, so lastly, I'm going to talk a little bit at how I looked at the human impact on these microbial communities. So I classified all of the pits as such. These ones are on the racetrack, and these ones were off the racetrack. And so then you can look at this all using a multivariate analysis, and the everything. So if you cut it off at 0 right here, every little dot represents a sequence, and all of these ones that are negative belong to one of those pits that were on the racetrack, whereas all of these ones up here are sequences from those pits that were off the racetrack. And so I did this using sequences from each layer. So I split, let me go back one. So I split all of the sequences from these pits up into layer 1, layer 2, and layer 3, and did the same thing. Layer 1 and 3 didn't have anything that was significant. So these ones in red right here are the only sequences that are significantly different from any sequences up here. So then all of these red dots right here indicate a sequence from some microbe that is similar to everything that is off the racetrack that isn't really on the racetrack. So then you can say, what are those sequences? And this isn't really that exciting. It's just Halobacteriaceae again, which is an archaeal sequence that was amplifying with our bacterial primers, which is kind of strange, but it's not always, it could just be that the bacterial primers aren't really acting in a domain-specific way. There's known issues with that. I have to look into this a little bit more and actually go in and pull out these exact sequences and see if where they line up to the primers and see if they're actually were hitting the primer or if it's some like weird sequencing error. Okay, so anyways, I don't have too much to say about that other than that we found a little bit of differences that might indicate human impact, and I'm going to look into it a little bit further, as well as do some of these other things, like make some more comparisons to, are there differences in these pits in mineralogy, elemental concentrations like heavy metals and sulfur. As I mentioned before, we're going to metagenomically sequence some of these samples to sort of get an idea of what genes are present and link that back to the incubation data. And then I have some additional headspace results that are going to come in from all of those incubations. And I'm going to do the Raman single-cell sorting of these samples. And then right now, or earlier this week, I guess, my lab was going out and sampling wells that are located on the salt flats. So we have shallow wells and deep wells, and we'll be sequencing those and sort of comparing them to the surface communities to see if we can see if some of the shallow wells in the surface communities share similarities. And with that, I would like to acknowledge my lab, the Brazelton lab, as well as our collaborators in the geology department, Brenda Bowen and Betsy Klebla, who was the lead microbiologist who was at the University of Westminster, but has since changed to sample-minded. Yeah, so thank you.
What kind of salt is on the salt flat? Is it just mostly sodium chloride, or?
I think it's got more magnesium than sodium in it. I think magnesium is more than potassium too.
Magnesium chloride?
Yeah.
Or?
Okay, real quick, I looked at, it looked like the soil below the salt was really dark. Is that true when you're there? Is it really dark? So
Yeah.
I mean, there's a lot of organics, I guess, that are below the salt.
Yeah, so I'm really excited to get some of the mineral data back from the geologists. They have an undergrad who's going to process all those all summer long for me. But yeah, some of these pits, you would dig down and you would hit this like weird interface between two salt layers that was just really black and sulfury smelling. Like as soon as you hit it, you were just like, oh God, that's sulfur.
Cool, all right, thank you.
All right, we have time for one last question.
So were you able to remove the community members that were predominantly present on the racetrack from the rest of the data?
I don't know if I understand. Let me go back here. Okay, so from all of these guys?
Right, because you would assume that those are due to human impacts?
Well, the problem with this is that none of these were really significantly different from ones that were off the racetrack. So it seemed like we had sequences that were different off the racetrack that weren't found on the racetrack. So it was the other way around.
Got it, okay, thanks.
All right, thank you very much.